The blood specimens come in a purple top tube. Downstairs, the micro CPA people, or MPA people, they put an aliquot in those tubes for us. They label the tubes and give us the label. Then we make a plate map. So the 96 well plate that will result with all of the extracts, then I will know what's in each well. And our CPA people downstairs, they look up the white counts. If the white count is high, we need to dilute the specimen because toward the end of this process, there's a plate that has filters in it, and it could clog in the filters if the white count is too much.
There's too much.
So that's what they do for us downstairs. They aliquot and give us the white count so we know if we need to dilute. And so we have the samples and the tubes ready to put on the instrument.
And the
when we
high
bring them
white
up.
count would be about 50, 20,000?
Anything over 12,
Anything over 12.
we dilute just out of trial and error. When we started with these instruments, we started seeing where it was. We started having clogs, and we just kind of figured out that that's about a good place to start diluting.
For the cutoff. Yeah. And the purple top tube is the one with the ed top, right? Is that
EDTA.
With the EDTA.
Yeah.
So it's is it the plasma that you get, or the whole?
We use whole blood.
You use whole blood.
Whole blood for this. Yeah, if you look at the tube, I don't know if you could. Well, actually, I have extra. So we get the whole blood. These are too many for today. I have to put these back for tomorrow's run. So they give us the whole blood. That's why we get the EDTA. We don't want it separated.
Got it.
I guess some labs use plasma, or I guess maybe more labs than not use plasma. But according to Dr. Yen, whole blood is really the best way.
Yes, that is what she
The best way
always mentions.
to do it.
Right. So for right now, we're waiting for the step where the machine inoculates.
Let's see. Right now,
Vials?
let me see if it's done anything yet. Okay, yeah, it's just starting. Right now, it just sounds like it's priming itself. Those are the pipetters, and I just hear it's probably got. I don't want to open this up because it'll beep at me, but there's some buffers and alcohol and water in here, and down below here there's also waste and water. It may have just been pipetting water through to prime itself. But what it's going to do, or what it's doing right now, is there's a little vial of protease solvent. So first it puts that enzyme solution into all of the wells. Then it will pipette all the blood specimens into this, I don't want to stick my hand in there, but this plate right there, the plastic block type plate, that's sitting on a heater. So that's the first step. It puts in the protease enzyme, the blood, and there's some heating, and so this is going to lyse the blood cells. And then as it goes on, it takes about 2.5 hours for the whole process till we get our extracts in the final plate. But there's a series of alcohol is added, there's wash buffers.
So all these reagents that would help lyse the white blood cells in order to release
and then it'll release the nucleic acids, and then to extract it. What does the technician do? Well, we load up the instrument with all the disposable. There's all these various plates that are part of this. There's reagents that go here, reagents in here, putting the specimens on, and then we just kind of let it go for the 2.5 hours, and then after the fact, after we're done, is when actually we have a lot of maintenance to do on this, because we have to wash all of. There's a lot of. Here, this one's not being used. It requires a lot of cleaning and maintenance, because with anything with DNA, you have to be you have to keep everything very clean, because you can easily contaminate. Obviously a little bit of DNA. We're going to amplify it in the end, so so any contamination could cause a huge problem.
Yeah.
So with anything to do with PCR and extraction, there's lots of cleaning involved. So most of our time spent with this instrument is not with the actual setting up it's after the fact, taking all of these pieces out. Everything comes out. Everything can be picked up. We have to clean. We have to wipe with alcohol. Some of them get soaked. We have to refill the reagents. We have to wash out the waste containers. So
And that's done
we
once
spend
a day.
Yeah. Oh yes. After every run, and then we have to refill all the pipette tips because there's a lot of pipetting. So there's a lot a lot of the stuff isn't high tech stuff. It's mostly loading it up, and then after we take it off, a lot of cleaning and refilling. So that's what takes the tech a lot of time here. It's mostly the maintenance and cleaning of the instrument more than once it's going. I mean, it's doing all the pipetting. So there's so now it's starting to pipette the blood samples. And on the screen, I can see where it is visually, but on the screen, I can see the blue one is the one that's pipetting. And after it pipettes into the plate, hopefully I will get all green squares there. If I get a red one, that means it's detected a clot. So it starts off in this block here. Like I said, there's enzymes in the blood and heating that goes on. And then you can see it has a plate inside of that large metal block. That is this type of plate. And this is the plate that has the filters on it. And that big metal block is sitting on top of a vacuum. And so later in the process, the samples from this block where it's going now are going to be pipetted into this filter plate, and it's going to be sucked through. And the nucleic acids should be on the filter, and there's going to be wash buffer that's going to go in there, get sucked through with the vacuum, and then later on, this taupe, it's an elute fluid, that'll get pipetted through the filters, and it will elute down into this plate, into these little tubes that are in that plate. So that's where the extract will end up, and it's just a clear fluid. We have to pipette from there into another plate that will go on to the ABI.